Students, today we are going to study about secondary metabolites and their production. Now, the secondary metabolites can be produced in the whole plant system, or we can produce it in the in vitro, that is in the laboratory. So, you should first understand the two terminologies what we are going to use in this. One is the primary metabolites and the other is the secondary metabolites. Now, the primary metabolites, these are molecules which have a physiological role in the process of plant survival. They are involved in both the growth and development of the plant system, and the secondary metabolites, they are derived from the primary metabolites. They are not involved in the growth and development process of the plant and are mostly produced under the condition of stress. That is, it can be abiotic as well as biotic stresses. But still, they have a multiple role in plants. They are produced as a source of protection in the plant. Apart from that, they have various application also, such as they can be used in the pharmaceuticals, in the feedstock, agriculture, flavor and fragrance production. They have antimicrobial activity. They can be used as food additives. So these secondary metabolites in totally, they can be classified into three main compounds, that is phenols, terpenes and nitrogen containing compounds. Again, each of these are again further classified. Now, if you look at the site of production of the secondary metabolites, it can be produced in the whole plant. Whole plant can produce the secondary metabolite, or it can be very localized. That is, it can be produced either in the flowers or in the leaves or even in the barks or even in the roots. So let us look once again into the definition. Secondary metabolites are generally defined as small organic molecules produced by an organism that are not essential for their growth, development, and reproduction. Secondary plant metabolites are numerous chemical compounds produced by the plant cell through metabolic pathways derived from the primary metabolic pathways. So now you know that they're having so much advantages and applications. So the question what arises is that how can we commercialize the production of the secondary metabolites? So there are two ways in which you can commercialize them. That is, first you can either produce it in the whole plant. We can use the whole plant as a factory, or we can do it in vitro culture. In vitro is in the laboratory. Now suppose say if you are using the whole plant system, we would either have to grow this plant in the field or we have to harvest them from the wild. Now you can see in the forest and all the plants and trees canopies growing, and we have identified so many pharmaceutically or biologically important products from those plants, okay, which are grown in the wild. We just harvest them, okay. So either you can do it in the field or harvest it from the wild. Now then this is a problem, because if you're going to grow the plants in the field, we would be using our land for producing the secondary metabolites, and we'll be unable to produce the food which is very essential for the entire population. It would not be a viable option, and if we are harvesting the plants from the wild, the perpetual harvesting, that is what we do, there is never ending harvesting, we keep on harvesting, okay. The harvesting of the plants from the wild would lead to their extinction. In addition, if the plants are growing in the field, or either we are harvesting them from while they would be exposed to a lot of unpredictable changes in the nature, and as a result the yield would vary. So in order to have a consistent yield, it would not be good option to opt for the first two methods. Okay, that is the wild, from harvesting from wild or growing it in the field. So in order to have a consistent, we are not going for that two methods, but instead of that we will opt for the second option, that is in vitro. Why in vitro? Because in vitro it can be grown continuously, and it is reliable source of secondary metabolite production because there is no season specificity. We can scale it up much as we want. There would not be any danger of crop failure. And in addition to this, there is production of some novel compounds which are otherwise not found in nature. But in vitro system also has certain drawbacks. Like most of the natural products, they have been substituted by the chemical analog. Now, whatever the natural products are there, usually that is replaced or they have been substituted, that is, they have been produced in the labs itself by synthetic method, because the cost of production is very higher. Okay, even when you're producing chemical analogs or synthetic product, the production cost is very high. The high value specialty compound produced is very less in quantity. So what we have to do is we should think about the commercialization of these plant, that is the secondary metabolites. So therefore the best option is the in vitro condition. So the cells which are under the culture, when we are growing it in the in vitro also, there are certain problems. Okay, we might also face here also the production cost when we are doing in vitro culturing. The cells which are under the culture, they undergo mutation in vitro, so they are not stable, that is, they are genetically unstable. We constantly need to have a pipeline of cells which are homogeneously producing secondary metabolites, so that they will be uniform genetically also. So now if you want, costs also should be less, there should not be mutation, it should be stable, it should be homogeneous. And if you want to produce all this, the best option is in vitro. So how are you going to do it in the in vitro? What all is required? So you can actually divide the process, okay, into two main types. So first what we are going to do is we are going to select an elite genotype which would be high secondary metabolite producer. So we are first going to choose a plant which is going to produce a elite genotype, a superior variety of the genotype which is going to produce a high secondary metabolite, because we are interested in producing the secondary metabolites. From where you are getting the secondary metabolites? I have taken the example of plant, and from plant we are producing the secondary metabolites, or it may be even the microorganisms. So if you are telling that you have to choose a plant which is having an elite genotype, how are you going to select that plant which is having that all the good quality by genotype method? What they do is, usually the scientists, they go for chemical profiling of the genotypes. After they have done the chemical profiling, they select genotypes which is a high producer. Thereafter, in the second stage, what they do will, they will initiate the culture using high concentration of oxen. So the first is you are going to select an elite genotype which would be a high secondary metabolite producer. and we are going to select which is the elite genotype by using chemical profiling methods. After you select the genotype which is a high producer, in second stage what you're going to do is you're going to initiate the cultures using a high concentration of oxen. And what is the oxen you're going to use? You're going to use the 2,4-dinitropenox phenoxyacetic acid. This is an in um, a type of oxen. So for that, what you have to do first is, because why we use 2,4-D is because we have studied that it is very much it produces what helps in the cell division. So we are going to culture our explant. Okay, explant from where? From this genotype. Okay, so you're going to culture the explant, which can be a leaf tissue or any tissue from which it is known that has a high concentration of particular secondary metabolites. So we induce the tissue to produce what? Callus. So once we have initiated the culture, we keep it in the idle condition. In the next stage, what we do is we go for cell suspension culture. So all these details, the selection of explant, the formation of calli and suspension culture, you have dealt in your tissue culture. You can also go back to my videos which in detail have spoken about all these. How you select the explant, how callus is formed, how we do the suspension culture. You can go for those plant tissue culture videos and you can have a glimpse about it how it is done. So after you have produced the callus, then what you do, you raise the cell suspension culture and you incubate the cell suspension culture for almost 3 weeks. Thereafter we are going to draw an inoculum. From where? From this we are going to draw the inoculum from the cell suspension culture, and you put it into a selection plate which would have all the same composition as your suspension culture, but this is selection plate is a semi solid form. Okay, so you put it in a semi solid form so that the individual cells can grow and form colonies. So suppose say, okay, if this is my petri plate, and what I'll be adding all the nutrition what you have added for the suspension culture. Only thing is your suspension culture is liquid, whereas this is semi solid. You have to add some amount of agar. Then what happens, you're you're going to take the cells from here. Okay, your callus, whichever is grown, single cells from the callus. When you put it into the suspension culture, you're going to get all single cells. And then what you're going to do, you put that single cells in the selection plate, and then you will see that some of the colonies are formed. Okay, the colonies will be produced. So individual cells can grow and form colonies. So you put it in the selection plate, which would have all the same composition as the suspension culture, but it will be a semi-solid form so that the individual cells can grow and form the colonies. The next stage would involve screening of the individual colonies. You screen this individual colonies. We are going to test which colony is a high producer of your secondary metabolites. From this, you have to screen which colony is going to produce a high secondary metabolites. So we are going to test which colony is a higher producer, because Why we do this is because we know in a plant body also there is genetic heterogeneity. Okay, if they are heterogenic, they would not be much useful to us. So once we select that we are going to move into the next phase, that is, we see that all most of our cells are in the homogeneous form. Okay, so once we have selected that we are going to move to the next phase, which is your biomass production and the optimization of the media. So here what we do is we are going to produce the biomass. What is your biomass? Here it is your single cells which you obtain from the plant. That is explant, callus, suspension culture, single cells you got. You have selected from the selection media. The next stage is the biomass production. We are going to optimize the media. So what we do here is we have to do media modification and we have to change the kind of the culture condition. Why means we have to stop the biomass production and then we should move next to the secondary metabolite production. So slightly we will change the condition media. That is, it stops the growth and starts producing your secondary metabolites. So you can see here, this is how your cultures grow. Okay, first you have the lag phase and then you have the log phase, or it is called as a exponential phase. It is also called as a tropophase, and this is the phase. In the tropophase is the one where your primary metabolites are produced. So you all know that the exponential phase is the highly dividing cells. Okay, they are actively dividing cells, and that is why they are producing the primary metabolites, which is essential for the growth and development of the plant. And whereas your secondary metabolites produce, they are produced in your stationary phase, or it is also called as the idiophase. You know that in secondary growth phase, what happens? The number of cells growing is equal to the number of cells that are dying. So it is this stage in the idiophase is what your secondary metabolites are going to produce. This is also one of the important questions in your competitive exam. They'll ask you which phase. Okay, they'll tell you whether it is topo phase or idio phase. So the choice is the, you have to mention it is as an idio phase. And so now what happens, you're going, after you have brought about the change in the media condition such that the primary metabolites are converted into the secondary metabolites, what you have to do is you have to go for the extraction. So the next stage is the extraction, because the secondary metabolites, they are produced mostly intracellularly. So we have to adopt a process or a mechanism so that it is released into the media. So suppose say um, if this is the cell and your secondary metabolites are produced within the cell, so you should have a mechanism so that it is released outside into the medium. It should not be present inside the cell. You have to have a mechanism to see that the secondary metabolites which are produced within the cells are produced into the medium. From there, you extract. Now, the factors affecting the secondary metabolite production in vitro condition is greatly affected by the culture condition. It can be observed that when the cells are growing actively at this point, okay, this is the where their cells are growing very actively. This is the same, that is your cell is growing, dividing, but what happens to your secondary metabolites? They are not produced here. Okay. On the other hand, here the cells are almost, they are decreasing in number, but your secondary metabolites is being produced. So these both are inversely proportional to each other. Okay, higher the cell division, lower is your secondary metabolite production. Lower the cell growth formation, higher is the secondary metabolite production. So we can say that the growth and the production of secondary metabolites, they are inversely related. So it takes place in the stationary phase. And what happens during your stationary phases? Your media is depleted, and as the result, the cell stops growing further, and it leads to what is known as cytodifferentiation. And there are induction of enzymes which lead to the secondary metabolite formation. So, your what is happening in your secondary metabolites? Why your cells are decreasing? Because there is no sufficient amount of nutrition, and also there is toxics being produced by your cells. Now, are the cells happy under these conditions? No, the cells will not be happy. See, when we are not providing you food, what happens? Even you feel that stress, right? Similarly, here the plants also are feeling stressed because they are not having sufficient amount of nutrients. At the same time, even this toxics is getting accumulated in the body. I mean, sorry, in the cells. So when there is a stress, only then you see that the secondary metabolites are produced. So the process can be divided into two phases. The first phase is the biomass production, and the second is the, see here, first phase is your biomass production, and second is the secondary metabolite production. So it is possible to produce large amount of biomass with an increase in the accumulation of the secondary metabolites. In the phase 1, that is your biomass production, in the phase 1, the culture conditions like your pH, temperature, light, etc. would be very different from the culture conditions that are required in your phase 2, which is the secondary metabolite production. The various factors which are affecting your stage 1. What is your stage 1? It is your biomass production. What is your biomass here? It can be either your microorganisms, or it can be even your single cells from your animals also, even plant cells also can be used. Okay. Usually, like we see, it is from the microbial and the plant cells. So, in the stage 1, which is the biomass production, the first factor which affects the production is the inoculum density. So, we are talking about the stage 1. What is your stage 1? Stage 1 is the production of the biomass. Who's my biomass? It can be my microorganisms, or herein I am giving the example of the plant. So, what are the factors which are influencing the formation of the biomass? You can see here, that is the first is the inoculum density. See, if you draw a pyramid, the first here, the topmost, what you can see is the inoculum density. So from where are you getting the inoculum? We are getting the inoculum, that is, we have previously raised the cell suspension culture, and that one we use it as a inoculum, that is to start the proliferation. So this is very critical, inoculum, because minimum inoculum density below which the cell or the organ growth will not be optimal. So this stage needs to be optimized, and it varies from species to species. So inoculum density, you cannot choose how much ever you want. There is a definite requirement how much the cell density should be, and this varies according to the species, what type of species you have chosen. So it varies from species to species. We need to optimize the fresh weight requirements of the inoculum per liter of the culture. The next parameter is the culture condition. This would involve both chemical and physical condition. This would include tissue culture medium and the physical conditions like the as I mentioned here, like light, temperature, humidity, pH. For example, the pH, optimal pH for you have to choose an optimal pH such that your cells can are actively dividing. Okay, and talking about the cultural condition, okay, you have to see that you get a maximum amount of biomass. So if you need a large amount of biomass, you need large number of cells to proliferate. Okay, so in order to achieve large number of cells to proliferate, what we do is we supplement the media with a high concentration of oxygen. And what is the oxygen we use? I told you we use the 2,4-D so that the cells divide actively and reach a particular density. So they let them proliferate. Okay, that is one of your cultural condition. You should have an optimal cell number. So the cells actively divide and they reach the particular density before they can be transferred to the next phase, which is your selection of your secondary metabolites. So in selection, what happens is um, again you can see there are various factors which can influence the selection part. Now, apart from this, before going to selection, even your temperature plays a very important role. Your media and the light. Okay, what is the you see that the cells would divide actively. Then the light also influences the cultural condition, and the temperature range is typically between 17 to 25 degrees centigrade. But actually, in your tissue cultures classes, you would have studied that your optimal temperature is 25 plus or minus 2. You can please go into those videos and look into the details. Okay. We say the optimal temperature growth in vitro culture is 25 plus or minus 2. But since you're producing here what you're producing secondary metabolites in vitro, the actual temperature what we use is the 17 to 25 degree centigrade. Now coming to the next factor, which is the selection of high yielding genotypes. Now we know that during the culture condition, there are many changes which happen to the cells or to the organs. The reason being the culture itself. Okay, what is bringing the change in the culture is the cell, the culture cell itself is bringing about these changes. Now these changes can be either like it could be a genetic variations, like it could be an addition or deletion of the chromosomes. So as a result, what is happening, a high producing cell line can get converted into an heterogeneous form because of the whatever the mutations which are occurring. Either addition can occur, deletion can occur. So whenever this occur, do you say it is homogeneous? No, they're not homogeneous, they're heterogeneous population. So where the secondary metabolite production will not be optimal when your plant of selection, the selection of your genotype is heterogeneous, you will not be getting an optimal production. So what you have to do, we need to make the lines homogeneous. Okay, whatever the cell lines you're using, you see that they are homogeneous, continuous. This can be brought about by a technique called as single cell cloning. So what are we going to do in this technique? Why we are going for single cell cloning? So that I get a homogeneous continuous cloning. growing cells. So here what we do is we will take an inoculum from our suspension culture, whatever the previous slide you saw. Okay, we will take an inoculum from the suspension culture plate and we plate it in a selection media, which is a solidified with gel, and we allow the cell to grow. The same previous slide what I told you here, we allow the cell to grow. Each of the colony would be screened for the secondary metabolite production. The ones which are high producers will thereafter would be multiplied and taken into for the production. So this is a continuous process which one needs to do. While doing so, you cannot do this in small scales. So you usually try to do in the large scale. That is, you're going for scaling up. What are you scaling up? You're scaling up your production of the secondary metabolites. Now, that is one of the third parameter. Now, coming to the next, the fourth parameter, that is your agitation. Now, agitation is another important parameter which controls the cell culture, and this is irrespective of the scale up operation. So, what is the importance of this agitation? It promotes the growth of the cell, and it brings about, that is, it makes the condition making the nutrients available to the cells, and also it sees that there is diffusion of gases in and out of the culture medium. It prevents the buildup of the carbon dioxide and other toxic exudates from the cells, and the media is sufficiently oxygenated. However, if your extreme agitation, if you're going for extreme agitation, this can have a deleterious effect, because when we are agitating or when we are shaking a culture at a very high speed, what is happening? The cells will get disintegrated. Okay, we told that we need a particular density of cells, and now when you're disintegrating your cell, the culture cells, this leads to the reduction in the size of the cell, which also is very important, and the cell viability will be compromised. So to prevent this, plant cells are often grown in stirred tank reactors, STIRRED, stirred tank reactors, at very low agitation. So this is about the agitation. So inoculum density, culture condition, selection of high yielding genotype is here, agitation is here. Next we'll go for the aerations. So usually we are doing agitation at low speed. So that is why we go for the stirred tank fermenters. So the next parameter is the aeration. It has three main functions. First is the maintenance of aerobic condition, and second is the desorption of volatile products which may be detrimental. So these volatile products, they are produced in their cells, because most of the cells, what happens is, um, when they produce these type of volatile compounds, it can cause an inhibition to the secondary metabolite production, and it may be detrimental. When we are providing aeration, it leads to the, so whenever the secondary metabolites are being produced, usually you see that the formation of volatile compounds. But if the volatile compounds are being produced, it will have a detrimental effect on your culture. So you have to see that there is no volatile compounds. So what does your aeration do? Your aeration, it sees, it causes the desorption of the volatile products. Okay, and then it helps to in the removal of the metabolic heat in certain cases. It has been observed that high concentration is toxic to the cell's metabolic activities, and it also decreases the nutrient availability from the culture broth. So aeration helps to eliminate all these problems. And as we know, the carbon dioxide is essential nutrient in the plant cell culture. So it also helps in the equal distribution of all required gases, whether your carbon dioxide or oxygen. It aeration will look into these features so that there is uniform distribution of all the gases. Now, this is till now whatever we spoke was about the production of the biomass. Okay, this step, biomass. That is, whatever the cells, what you're using, what are the factors which are necessary for the biomass production. Only when you have the biomass production, you can, from this biomass, you can produce the secondary metabolite. That is, in other words, from the cell lines or plant cell lines or your microbial cell lines. Only from that I can produce the secondary metabolites. So these all parameters are for your primary, production of biomass. Next, we will see that what is required for the secondary metabolites production.